Good morning, everyone. Thank you to all those who have joined us and welcome. My name is Lisa McCoy. I'm the CEO for Defeat Duchenne Canada. On behalf of our organization, I want to welcome you all here this morning for this very exciting and informative presentation with our friends from PepGen. Before we get started, I'll just share a little bit of our organization, and then I'll hand it off to our wonderful presenters. Defeat Duchenne Canada is our country's only charity that is dedicated to ending Duchenne muscular dystrophy. So obviously, we're very excited about this presentation today, as I'm sure all of you are. Our mission involves providing education and support, advocacy, and importantly, funding innovative, world-renowned research for Duchenne muscular dystrophy. Of course, none of that would be possible without the generosity of our wonderful community of donors. We rely very much on individuals, our Duchenne family, our community partners, corporations, who help us by donating their time and their money to support all of the activities that we do. And they do this in a variety of different ways, which you can see on the slide. We have many different options for people to support our organization, and we truly wouldn't be able to do it without them and without you. So thank you so much for your support. So without further ado, it's my great pleasure to have the opportunity to introduce you to today's presenters. Let me start first with Dr. McMillan, who is a world-renowned and a very highly respected clinical investigator for the Children's Hospital of Eastern Ontario Research Institute. Dr. McMillan has been in the area of clinical and translational research in pediatrics, neurology, and neuromuscular medicine for many years. He has been a principal investigator, and as you can see from his bio, we are very honored to have him with us today. And joining him from PepGen in their patient advocacy is Elena Tress. Elena has many years of experience in patient advocacy and medical affairs. She's a highly qualified individual, and we thank her very much for being with us today. So without further ado, let me hand it over to Dr. McMillan, who's going to start the presentation today.
Thank you. And this is Elena speaking. Before Dr. McMillan begins, I really wanted to give everyone an overview of who PepGen is and what we learned so far from the community. I just want to make sure, can I get a thumbs up from someone that you can see my screen okay? Okay. So we'll be talking today about our PGN EDO 51 program for Duchenne muscular dystrophy. I wanted to start off with sort of an about us slide about PepGen. So our mission is that we're committed to changing the lives of people living with neuromuscular and neurological disorders. We are based in Boston, Massachusetts in the USA, so that we're sort of in the upper right-hand corner of the US. We're small but mighty. We're just over 60 employees. And what you'll hear us talk a lot about today is how we study investigative enhanced delivery oligonucleotides. That's a very long word, so we call them EDOs, for the potential treatment of Duchenne muscular dystrophy and myotonic dystrophy type 1. And right now in the clinic, we have a study for myotonic dystrophy type 1, but you'll be hearing a lot about today about the other program we have in the clinic, PGN EDO 51, for the potential treatment of DMD. And on the right-hand side here is a photo of one of the community families that we've worked with closely that do live fairly close to the office, and we've had the privilege of meeting in person. The agenda today, I'll take you through our commitment to the community, sort of how we've worked with you and what we've learned from you so far. And then the rest of today will be Dr. McMillan walking you through those EDOs, our preclinical program data about PGN EDO51, and then the study we have open, otherwise known as Connect One, and then there will be an open Q&A session. Disclaimers, there's a lot of information on this slide. I think the biggest takeaway for today from here is just to know that everything we're talking about is investigative. Nothing we are talking about today has been approved for commercial use by any regulatory agency. If anyone has any questions on anything about that, please feel free to go to our website. And so narrowing in on our mission, we are really seeking to utilize our EDOs for the potential treatment of transforming the lives of people living with DMD. These are some other wonderful community members we've been able to engage with, Mallory and Colin, who have really taught us a lot along the way. And before we try to further educate you on our technology and our data and our Connect One program, I want to stop and take some time to thank you and show you how much you've taught us and how much we've learned from you. So there's been some key community insights that we've gathered along this PepGen journey so far, especially around designing our clinical trials. So what we measure in our clinical trials, what are things important to families living with DMD, and how do we measure them to see if we have an impact, and also make sure that their time is spent wisely joining us in a trial. What a visit to the clinic looks like for our trial. Joining any clinical trial is a huge commitment. There can be a lot of challenges, and we've heard a lot from the community how to structure the days in clinics, order of assessments, schedule of assessments to try to minimize that burden and keep in mind things like fatigue and boredom and anything else that could come from a clinic visit. We're trying to make this as comfortable as possible. Another thing we've heard a lot about and have gotten a lot of tips and tricks from the community on is travel. For most people, a clinical trial is not happening nearby. So if it's not, what are ways to make travel easier, coordinating it with families and the clinical study staff, also prepaying for that travel and having that reimbursed in a way that families aren't further burdened with, bureaucracy and trying to get a flight booked for them or a flight reimbursed. That's something we really worked out with our vendor on that we're very proud of. And the materials we put together. So we spend a lot of time at PepGen thinking about how can we meet the community where they are and inform them of the things we're talking about today, like our preclinical data, our mechanism of action, what we think is important to know about the study. And that's a lot of feedback we've gotten. And this last piece is so important. It's not a bullet, it has its own ellipsis, is the entire drug development process. A clinical trial is just one part of otherwise a very long process and journey, if you will, that you guys have partnered with us on. And we are so thankful for your insight, your ideas, and it's always a privilege to hear that from you. And on the right-hand side, sort of as a thank you to all of you, this is a photo of our staff taken about a year ago. So we're bigger now. I think we're roughly half the size last year that we are right now, again, with that short-lived family. And there are so many people here at PepGen that are so thankful for you with so many different backgrounds and expertise that they've had to partner with you on this process. And some examples of how we've engaged with you, I just want to leave you with this. So these are really how we learned so much from you. So one, building connections by in-person engagement. That is a photo of a science camp we did earlier this year with some local DMD families. And in that photo is one of our scientists, John, working with a young scientist there on some litmus paper testing of some household items. And we're always so thankful just to have that fun and that quality time with people of the community. Then we have a very humble screenshot of good old Microsoft Outlook. We manage numerous channels to make sure that we have open communication channels with you if we can't be in person. We have the email inbox community@pepgen.com, where we always look forward to any questions you have, feedback you have. If, we will make sure that someone has an answer to your question and get back to you there. The middle of the screen here is the materials that I mentioned. This is a screenshot of one of the handouts we typically give at conferences that give an overview of many of the things you'll hear about today. And we've gotten some really great feedback from people in the community on how to explain them, how to optimize the layout, and just what matters most to people of the community. And meeting you at those conferences, we know that it's a heavy lift to go to a conference. They're long days, they're tiring days, but anytime that you guys take a moment out of your day to come join us at our table or say hi to us at the buffet, whatever it is, we're so grateful for that. And it helps us just get to know you better and serve you better. And last but not least, advisory boards. We've had the privilege of hosting numerous advisory boards with the Duchenne community of people of different ages, different backgrounds, different geographic areas, different stages and phases in their own Duchenne journey. And we've gotten some really incredible feedback on our clinical trial design. And it's really an opportunity for the community to sort of look under the hood of PepGen, so to speak, and look at what we're doing and how we're doing it. And we're so thankful for all this. And we just wanted to share those examples with you. So I am now going to hand it over to Dr. McMillan, who is going to give you the rest of the exciting updates for today.
That's great. Well, thank you very much, Elena. Thank you to Defeat Duchenne Canada for the opportunity to speak today and for all of the very important work that you're doing. And to PepGen as well for moving this very promising treatment forward and for also choosing to run this important clinical trial in Canada. And I can say as a Canadian investigator, as a Canadian clinician, it's very, very important that our patients have access to these emerging therapies and all of the excitement and the hope that they bring with them. Next slide, please. So I do have a few disclosures from advisory boards that I've assisted with, and I'm also a member of a data safety monitoring committee for a gene replacement therapy that does not have anything to do with Duchenne muscular dystrophy. Next slide, please. So one of the places that I want to start is to talk a little bit about something that's called the reading frame hypothesis. And I'm going to make this very accessible so that it can be understood by those in the audience that don't have a PhD after their name. One of the first places that I like to start is to remind people the differences between DNA, RNA, and proteins. So DNA is our master blueprint that lies within our body's cells. We have one copy of these master blueprints within each cell. And it's precious. It stays in a very, very important part of the cell called the nucleus to protect it so that it doesn't get damaged because it's so critical. Now, RNA is a product that's made from DNA. And the analogy that I would use is if DNA is the master blueprint, RNA is the photocopies of that master blueprint that are brought out to the construction site so that it can be used in order to build the buildings, to make things run. So that RNA is critical that it gets assembled properly in order for it to do its job on the construction site. And the protein, like dystrophin, are like the workers. They carry out the very important role. But each of those steps are very important along the way for the job to get done. And in this case, for muscles to remain strong. So if we look now at the mad cat ate the big rat and the fat bat, what that is that's looking at the DNA. And this is a normal situation where there's no problems associated with it. Our body is able to read that, to make the photocopies, the RNA, and then turn that into a functional dystrophin protein that you can see in the middle column, where there's this nice red ring around each of those muscle fibers. Now we run into a problem. So with DMD, it doesn't matter whether it's due to an exon deletion where part of it's missing. So in this case, if we consider a very, very small deletion where the TH are missing, or if there's a duplication, or if there's even just a switch of some of the letters. But then the net is that because of that, in this case, deletion, it now no longer makes sense. So when things are getting made into RNA and things are getting read to make proteins, our body reads them in little chunks of 3. So because there's 2 letters missing, now it no longer makes sense. It would read something like the mad cat 8, ibiga, gra, tan, ditha, ifa, tiba. It doesn't make any sense anymore. And that's the issue with Duchenne muscular dystrophy. And like many other diseases. And because of that, the RNA gets broken down and there's no dystrophin. There's no protein that's made. Now, if we consider an exon skipping therapy, it's a specific oligonucleotide that binds to a part of the RNA, to those photocopies, and allows it to get better made into protein. So in this case, there's still the mistake. That doesn't change. But these oligonucleotide is binding to the RNA in such a way that it's silencing or cutting out that remaining part and allowing things to be read properly. In the case of deletions, for the exons on either side to be able to be fit together. And in this example here, it's now changed things from that completely incomprehensible gibberish to the mad cat ate the fat bat. So it now has meaning. It's not exactly the same as the original. It's shorter, but it is able to do its job partially and restore that reading frame to allow, in this case, some dystrophin to be made. And that's the key here. I'll ask for the next slide, please. So the goal of these antisense oligonucleotides is to take a situation like Duchenne muscular dystrophy where there's no dystrophin made and convert it into a milder form, which we refer to as clinicians as Becker muscular dystrophy. So it's changing that phenotype to a milder phenotype by allowing some partial length dystrophin to be made. As I showed you in that slide, the antisense oligonucleotides are designed to allow skipping over of part of the transcript, restoring the reading frame, and allowing it to now make sense again. There are several ASOs that are approved by the FDA in the United States, and that includes eteplirsen, which is exon 51 skipping, golodirsen and vitilarsen, which are exon 53 skipping, as well as casimersen, or exon 45 skipping. And there have been a number of Canadian sites that have been involved in these trials as well, including Ottawa. These therapies need to be administered every week by ongoing intravenous infusions. But you'll see a little bit later on, because of some of the advances, now the dosing therapy range is starting to extend to every month. And that's allowing it to be even better tolerated in some ways, in addition to it being more effective. Now, if we look at the antisense oligonucleotides, the ones that are currently approved in the United States could theoretically benefit about 30% of patients with Duchenne muscular dystrophy with exon deletions. And if companies were able to make antisense oligonucleotides, they could potentially benefit 79% of patients with exon deletions. So if all of the potential ASOs possible were made, up to 79 patients with deletions could be helped by these ASOs. Next slide, please. One of the biggest challenges that has been faced with the antisense oligonucleotides that have already been studied and have been approved is on the left. If we're seeing this oligonucleotide on the left, which we tend to refer to as naked, meaning that there's nothing that's attached to it or conjugated to it, is that there's a relatively smaller proportion of it is able to get into the cell. So we can see here where the purple X is that the antisense oligonucleotide, some of it is able to get into the cell and into the cytoplasm to have its effect on the RNA, those photocopies, but not all of it can. One of the really important things with conjugating, in this case, a peptide to the antisense oligonucleotide, is to improve the penetration into the cell. So if more of it is able to get into the cytoplasm, then more of it is able to do the job of restoring that reading frame and allowing much more of the partial length dystrophin to be produced. And we're seeing that on the right. Much more of those oligonucleotides are getting into the cell to do their job. Next slide, please. What we see here on the left is we see the ASO, or the antisense oligonucleotide, as this long gray, almost spiral type shape, and that's what's going to be binding to the RNA. And then at the very top of that, the darker and light blue color is the peptide that's bound to it that allows for better penetration into tissues, including skeletal muscle, and also cardiac and smooth muscle tissue. Next slide, please. Like any investigational product, any emerging therapy, it's studied in a stepwise manner, from initially looking at cells from people who have DMD to looking at mouse models like the MDX mouse, to then moving up and looking at higher mammals such as non-human primates, and then eventually healthy volunteers before, as is the case with PepGen's EDO51, moving into clinical trials for people who have DMD. We see on the two columns on the left, from the work that's been done in cells and in mice, that there's very, very effective delivery of EDO. There's enhanced exon skipping and a significant improvement in dystrophin production as a result of that. That was also confirmed, the delivery in the non-human primates and in the healthy volunteers. And in addition, with those other targets, the monkeys and the humans, it's also been demonstrated to have a very favorable side effect profile in those populations. We can't comment on dystrophin production in monkeys and in healthy human volunteers, because neither of those two on the right have DMD. So it's been proven in the cells, it's been proven in the mouse to improve dystrophin, and it's showing a very favorable side effect profile in the mice, the non-human primates, and in the humans. Next slide, please. What we see in this slide is this is some of the earlier preclinical work that was done on mice. And we see on the left a naked oligonucleotide, antisense oligonucleotide. And in comparison, when there's a protein that's conjugated to it, a significant improvement in the exon skipping, because more of it's able to get into the cell. In the mice, I should note that they use an exon 23 skipping variant, and it's just because the gene is a bit different, and that needs to be the target in mice, but it's entirely equivalent to exon 51 skipping. So much better penetration, very, very encouraging in the mouse model. Next slide, please. The other thing we see here, the work that was done on the mouse, are two things. So on the middle column that begins dystrophin and then in brackets Western blot, that's the quantity of dystrophin that we're seeing expressed in those mice. And what we see is that not only is the quantity of dystrophin improved, it continues to improve with repeat dosing. So on the very, very left, the large white box, that's showing a wild type or a normal mouse. The one after that is the MDX mouse. So that's the mouse who has the equivalent of Duchenne muscular dystrophy. And then the two on the right are those MDX mice that have been treated with either a single dose of EDO23 or 4 doses of EDO23, and a significant improvement with repeat dosing. The column on the right, dystrophin positive fibers, that's when the muscle fibers are stained with an immunofluorescence that reacts to dystrophin, and then somebody counts the number of fibers that are showing evidence of dystrophin expression. And that's very important for me as a clinician, because it's one thing for people to talk about dystrophin and there being more dystrophin in the muscle, but as a clinician and a scientist, I need to ask myself, is it getting to where it needs to be? Is it getting into the muscle membrane, which is very, very important. Next slide, please. The other piece that was very important, you'll remember when I talked about some of the approved ASOs that are given every weekly, is with EDO51, when it's been delivered to humans, it shows that it continues to have a very good expression even 4 weeks after the administration. And that allows it to only need to be given every 4 weeks instead of the naked or the traditional ASOs, which are administered every week. And you can see here at the for the 3 different dosing cohorts, 5 milligrams per kilogram, 10, and 15, there's an increase amount with the higher doses. And the result of dystrophin at day 28 or 4 weeks is the same, if not even a little bit higher than what it was 10 days after the medication was administered. Next slide, please. So as part of that, and I alluded to this, is we've moved from studies that have been done in cells to studies that have been done in mice and studies that have been done in non-human primates or monkeys, collectively referred to as preclinical work, to phase 1 studies that have been done in healthy human volunteers to where we are now. The clinical trial that's being run for boys and young men who have Duchenne muscular dystrophy. And we're one of the 4 Canadian sites that are part of the Connect-1 study that is being run to look at this emerging therapy in boys and young men with Duchenne muscular dystrophy. It's an open label study, meaning that each of the boys who are treated in this study will receive the medication. There is no placebo group as part of this clinical trial. Connect-2 will also be run in other countries as well, including the United States. But Connect-1 is ongoing. Next slide, please. On the left, we're showing the Connect-1 and Connect-2 studies. Connect-1, again, run in Canada, ongoing now. PepGen also has additional preclinical work that's being done in other exon skipping therapies. So exon deletions that are amenable to exon 53 skipping, exon 45 skipping, and exon 44 skipping. So this is additional preclinical work that's ongoing right now. Next slide, please. Now I'm going to focus a bit more and just provide some details about the ongoing Connect-1 study. Next slide, please. When clinical trials are designed, there's a lot of different inputs and information that is used to help to design those trials, to make sure that they're run appropriately, sensitively, and they're designed to answer the questions that are necessary around safety and potential efficacy. And that includes looking at data that's available from registries. In Canada, we have the CNDR, the Canadian Neuromuscular Disease Registry, a very, very important resource for us to learn more about DMD. Patient advocacy groups, so really important that we turn to the patients themselves and to families to understand what matters to them so that the correct things can be measured and the trials can be designed in a sensitive way. Community advisors and ad boards, as well as turning to people like myself, clinical advisors and clinicians, for us to be able to share our experiences as well. And all of that is put together, and then a team will design a clinical study, again, for it to be run to answer the questions regarding safety and potential efficacy of a treatment. Next slide, please. In the case of Connect-1, travel and feedback, there is custom travel arrangements that are provided by a travel concierge that is contracted by PepGen. And participants, patients and their families are able to provide feedback about that, as well as their study experience overall. There is some age-specific materials that are provided in the form of information sheets, so people can understand more about the procedures themselves and what they entail. Certainly, that's not a replacement from the discussions that should be taking place between the principal investigator, like myself, and the patient and their families for them to understand. But a very, very important adjunctive source of information. And there's a participant planner, so the families will have upfront details about the study itself. And then on the right, there's DMD-specific training for all of the study team members. The site staff are trained to explain the study to people that are from a wide range of different educational, different cultural and ethnic backgrounds. And then there also needs to be a quiet time and space in the clinic, both for people to have time to make the decision around participating in the trial, as well as for those who are moving forward and participating in the trial, for them to have breaks in between some of the clinical evaluation that's carried out. Next slide, please. The protocol title, and it's always a long one, but it's a phase 2 open label, multiple ascending dose of PGN-EDO51 in participants with Duchenne muscular dystrophy that are amenable to exon 51 skipping. That's the CONNECT-1. And the multiple ascending dose, I'll show you on the next slide, is because as each cohort closes and is deemed by a data safety monitoring committee as being safe, then the next cohort at a higher dose will open and the same process will be repeated. Next slide, please. The purpose of the study is to evaluate the safety and the tolerability of repeat dosing of the study medication PGN-EDO51 that are administered to people with Duchenne muscular dystrophy. And as I mentioned before, the design of this study is an open label study in ambulatory and non-ambulatory people who are living with DMD and who have a mutation or a pathogenic variant, a deletion that is amenable to exon 51 skipping. The endpoints are safety and tolerability, the amount of dystrophin that's produced in the muscle, the exon skipping that's induced, participant questionnaires, and then various measurements of function. And we call those timed functional test results, such as how fast or how quickly someone can walk 10 meters, for example, as well as measuring the drug levels in the blood and in the urine at various time points. And this is also very important for myself as a clinician, because this is a study that not only includes ambulatory boys and young men, but also non-ambulatory young boys and young men, a population that's often been excluded from clinical trials that have been run in the past. Next slide, please. Canada is the sole country that CONNECT-1 is being run in. There are 4 different sites, as I mentioned to you before. There's my site here in Ottawa at the Children's Hospital of Eastern Ontario. Dr. Gunarowski is the principal investigator in Toronto at Sick Kids. Dr. Nicolas Chrétien is the principal investigator in CHUL in Quebec City. And then Dr. Colleen O'Donnell is the investigator at New Horizons Health in Fredericton, New Brunswick. The participation in this trial, the study itself will run for 4 months, and then there is the long-term extension that runs after that, where people will continue to get access to therapy, and the entire participation itself could be 2.5 years or longer. There's a screening phase that's done where the investigator and the team will do various tests, blood tests, timed function tests, to be able to make sure that the boy or the young man meets all of the criteria for them to be eligible for this clinical trial. The study assessments require 2 muscle biopsies to be performed. This is something that is increasingly being done with a number of different clinical trials. Not only antisense oligonucleotides, but also gene replacement therapies. One is done as a baseline, and then another one is done at week 13. There's blood and urine tests that are done periodically. There's an ECG and an echocardiogram, various heart tests. There's breathing tests known as spirometry or pulmonary function tests, and then there's questionnaires to be completed as well. And as I mentioned, for those who complete the initial phase, there's the long-term extension study that is carried on after that to ensure access to this therapy, A, until the trial is completed, and B, until regulatory approval and commercial access becomes available. Next slide, please. This is a figure or a slide that's showing you the design of the study. You can see in the blue bar on the left, the dark blue is when the screening takes place. Then on the very bottom is the initial cohort, 5 milligram per kilogram dosing. There's the biopsy and then the 4 different doses, a repeat biopsy. And then the Data Safety Monitoring Committee will decide after there's 2 doses if the principal investigators can then move to the next cohort, 10 milligrams per kilogram, to begin the same process again and be dosing boys and young men with the 10 milligram per kilogram dosing. And same process after all the participants have had 2 infusions of that, they will meet and they will tell us if we're able to increase further to a higher dose after that. Next slide, please. The key inclusion and exclusion criteria. So these are the criteria that one on the left needs to have to be part of the trial and can't have on the right in order to be eligible for participation in the trial. So the inclusion criteria is that one must be a male who is at least 8 years old, can be ambulatory or non-ambulatory, must have a confirmed diagnosis of Duchenne muscular dystrophy, and must have a deletion that's amenable to exon 51 skipping. They must weigh at least 25 kilograms, and they must have enough strength in their arm to be able to lift a 200-gram cup up to their mouth. The key exclusion criteria is any other clinically significant condition or medical condition that would interfere with the ability to perform study assessments, any prior treatment with gene replacement therapy, any infection in the past 2 weeks, or infection requiring IV antibiotics within 4 weeks, and any surgery or anesthesia that's within 3 months, or expected surgery requiring anesthesia that's predicted during the study duration. Next slide, please. And on that note, I'll close, and there's lots of time left for questions. I'd like to thank the clinical study itself, and in particular for giving an opportunity for this to be carried out in Canada, for our Canadian patients and families to be part of this important clinical trial. To the community broadly, and all of the clinical advisors that have been tapped in order to make sure that this has the optimal study design, as well as the clinical site staff, both at mine as well as all of the clinical sites, for their dedication, their hard work, and for moving this forward, making it available for our patients and their families. So I'll stop there. Thank you again.
Well, thank you so much. That was an extremely helpful and informative presentation, as we knew it would be. At this point, I wonder if there are any questions that have come in through our Q&A, or any that we may have received prior to the presentation that either you, Dr. McMillan, or Elena might be able to address. I'll turn it over to you, Elena, to see if you have some. Thank you.
Thank you. I know I've received 2 so far, so I'm going to go through those first, and then I'm also keeping an eye out in the Q&A box function of this Zoom if anyone else has any additional questions. So the first question I got was if in this study, or in the long-term extension period of the study, if there's any opportunity for home infusions.
Thank you for that question. The answer to that is yes. There certainly is an opportunity for home infusions to be given at one point during the 4-month study. But more importantly, at the end of the study, after the 4 weeks have been administered, the boys can receive the intravenous infusions after the 4 months have been finished, for them to continue to get access to it. At our particular site in Ottawa, all of the infusions during the 4 months of the study will be given either in hospital as an inpatient, really to make life easier, because we have to do some PK sampling where we draw back a little bit of blood from an IV line. So an IV goes in, so there's no need for repeat pokes. but blood is just drawn back at a few time points. And we need somebody to be in hospital because it's hard to collect blood from that saline lock or that IV line, or collect urine at 11 at night or 2 in the morning. It's, just so that we can do that. Other infusions at our site will be given in the medical day unit on an outpatient basis for those 4 weeks. But then after that, certainly an opportunity for the infusion to be at home, particularly important for those people who are traveling longer distances.
Okay, thank you. And then this was a question that was received this morning from Defeat Duchenne, and it's, would PepGen consider adding a single exon 51 duplication patient to the trial or a future trial? And it looks like they list a reference here, and that exon skipping may benefit single exon duplication patients.
Thank you again. The best way I can answer that question in the context of the current clinical trial that's ongoing is this specific clinical trial is designed for people that have exon deletions that are amenable to exon 51 skipping. So for this specific clinical trial, it would not be possible for somebody with a duplication to be part of it. Part of the rationale for that is that we acknowledge that no two people are the same, and that there is certainly variability from person to person. But when the clinical trials are being designed, they have to be similar enough that there's a greater likelihood that we'll be able to answer the questions that are necessary from a safety standpoint and efficacy standpoint for us to know if this therapy is effective for people with that specific mutation causing DMD. Now, the hope is, and I touched on this with sharing a bit about the pipeline in the future, is that if this is successful, and I certainly hope as a principal investigator that it is successful, the hope is that in the future, there will be the potential for there to be an increasing number of ASOs that are conjugated to various types of proteins or peptides for better penetration that will treat other exon deletions and potentially even exon duplications. So it's a very, very important question that I am optimistic that we will be able to look to in the future.
Elena, any other questions that you have? Because I have a few here that have come in. We have a couple in the Q&A that I haven't had a chance to look at, but I will. But we had a couple of other questions. Some of this stuff I know too, the people are saying you dealt with, but I think you're just looking for some clarification, Dr. McMillan. By the way, I do want to say how pleased we are to have been involved in working with you and the sites and in getting these trials to Canada is huge, and it's so important to the families for access. So, Elena, to you and your team, and Dr. McMillan, you and your team, I just want to express on behalf of Duchenne Canada our thanks for that. The question is, you mentioned a few things related to nucleotides. The question is, how much do we need to get into the cells to see a significant improvement, or normal development, the penetration percentage, etc.?
Part of the reason for this trial and others that are being run is to answer that exact question. So I think that's part of what we're hoping to be able to have more information with moving forward. I can say on a very basic level that there needs to be enough penetration for us to be able to see the exon skipping and the dystrophin production for it to have a meaningful effect on stabilizing the muscle membranes. So, whether the exact number is 20% dystrophin expression or 30% dystrophin expression, different people will quote different thresholds and will argue very adamantly as to why they think they are right. But I think as part of this clinical trial, and others like it, it's to answer the question of what sort of dystrophin expression we're seeing, how is that impacting our timed function tests in terms of how quickly people can walk 10 meters, how people are able to lift their arms in the air for those that are non-ambulatory, and very importantly, how that changes over time. That is ultimately what's going to be very important.
Thank you. Next question was, you mentioned the increase in dosing as the trial moves through each stage. question is about repeat dosing. And again, you may have already just answered this. It may be what you're trying to achieve, but what type of dosing frequency is expected to be needed in the human trials?
So the PGN 051 will be administered every 4 weeks, and that will continue as part of the trial and then into the open label extension phase. The reason for the dose escalation as part of this clinical trial is because it's important to start at the lowest dose to answer questions around safety. It's really important as a clinician that we always provide a dose, whether it's through clinical trial design and input we give on that, whether it's through medication that we're offering clinically, that we give a dose that is going to be able to offer some clinical benefit, but at the same time is not going to be associated with excessive side effects. So that's really the balance for that. So an ascending dose study design, like the one that's part of this study, starts at a lower dose, 5 milligrams per kilogram, to ensure that there's no side effects that are emerging, and a side effect profile, for example, and then it will move up to 10 milligrams per kilogram. And one thing that's really important is that the sponsor has been clear is that as we move up with the dosing, if the higher doses are also shown to be safe, then the individuals who are on the lower dose will be moved up. So they will not be kept on a lower dose that potentially might have a bit less efficacy associated with it. They'll be given an opportunity to move up to the higher dose once that question around safety has been answered.
Great. Dr. McMillan, thank you for that. And Elena is going to answer the next series of questions that have come through our Q&A. And I think that the last question that I had is going to be answered in there. If it isn't, perhaps you could include it in, and it's simply there was a slide they said, but what other exons were you working on? They said 45 and a question mark around some others. And that's it for me, and I'll turn it over to Elena.
I can let Dr. McMillan answer that if that's okay.
Could I ask you, Rick, just to if you could just rephrase that question for me, please?
Yeah,
just
it was there was a slide apparently that showed other exons that were going to be incorporated, and it just wanted to be sure what those were. I think 45 was one of them.
Thank
you. I didn't get what the other ones were.
Yeah.
Thanks.
So the classic ones, and the reason why these tend to be included, sort of, you could say, the next phase of exon skipping therapies, are exon skipping therapies for people with deletions that are amenable to exon 45 skipping, exon 44 skipping, and exon 53 skipping. And there are others. There are many others that can be produced as well. But those others that I haven't mentioned tend to be associated with a lower frequency of exon deletions that are occurring in people who have DMD. So they potentially could be made in the future, but attention would be pointed towards those less common deletion amenable mutations like duplications, once proof of concept has been proven, and we know with certainty that this is a therapy that is offering clinical benefit.
Great, thank you. And Elena, over to you, because I now see you've got a bunch of questions to answer.
I have a couple. Yeah, so I'm going to read the questions received to you, Dr. McMillan. So we got a question that is the current and second/third generation skipping more beneficial for boys with a single deletion or multiple exon deletions?
It has less to do with the number of exon deletions, and it has a bit more to do with the pattern. So for example, one of the analogies that's sometimes used is if we imagine all of the exons to be like puzzle pieces, and all of those puzzle pieces have to fit together in a line for the RNA to be made into the protein. So for those photocopies on the construction sites to be able to be interpretable. So sometimes if a person's missing, for example, exon 50, that's an exon 51 amenable mutation. Or if they're missing 49 and 50, so is that. Or 48 to 50, so is that. because the pieces on 52 can fit together with a few of the exons on the other end. But if somebody has a deletion in a different area, it may only involve one exon, but that would be something that would not be amenable to exon 51 skipping, and might be difficult to replace. Really, really large deletions that might involve, say, 30 or 35 exons, that also would be very difficult. But to summarize and to make it very clear, it has more to do with the pattern of deletion rather than the specific number of exons that are deleted per se. And you can Google it. There's a number of very good images on the internet. If you type in exon DMD, exon deletions amenable to exon skipping, and you can start to see exon deletion patterns that would potentially be amenable to treatment with exon 51 skipping, or 53 or 45, or some of the others that are at an earlier stage of development.
Thanks, Dr. McMillan. And then we got the question that if someone does not meet all of the inclusion criteria, strength as an example, when would they be able to benefit from the study?
Yeah, thank you. It depends on the specific inclusion exclusion criteria. So there are some of them, for example, if an individual has an infection a week before screening, they wouldn't meet the criteria, but they potentially could be re-screened, as time goes on. And then there are other criteria. For example, if someone has an exon deletion that's not amenable to exon 51 skipping therapy, then this would not be a treatment that would be effective for them. That individual would not be eligible for this trial because it would be giving someone a product that would not have any benefit and potentially might have some tolerability issues. Now, in the specific example that was given, if someone doesn't meet the criteria because they are either too young or because they are too weak, examples like that, they may be able to be part of a different trial at a different date. They may be able to access the therapy if and when it's approved by Health Canada and becomes commercially available. And I know one of the things that I'm certainly a big advocate for, and I know Duchenne Canada has been a big advocate for, is when there are promising therapies, particularly those that are approved in other countries, commonly the United States, we all need to do a better job of advocating and trying to get access to those products faster in Canada. So people like this individual that was mentioned, who potentially could be a bit too weak to meet the criteria for the upper limbs, if this treatment is shown to be effective, and if a company like PepGen is moving forward to seek approval in the United States, we also want to see companies seek approval with Health Canada so that it can be available for our patients as well. So, but commercial approval would ultimately be the final way to be able to access that for all individuals who had deletion amenable to exon 51 skipping.
Thank you. And we got another question around inclusion criteria on why is the trial for boys from 8 years old and not younger? Are we intending to include toddlers at some point?
I can say with regards to, again, this specific trial, that the decision was made by the company to limit it to boys that are 8 years of age and above. I don't have privy to exactly why that decision was made. It could be, and I would hypothesize that it might be because we want to be able to administer it to older children that are better able to explain maybe symptoms that they have, that have larger muscles, that are more amenable to biopsy, that are better able to tolerate and cooperate with some of the timed functional testing, like the ability to do a 10-minute walk or other tests like that and cooperate with it. But regardless of those hypotheses that I'm purely making, and I'm not suggesting that those were any of the decision-making processes that PepGen chose, the decision was made at this time for the lower limit to be 8 years old. The hope is, and we see this in many other clinical trials from other sponsors, is that when that critical age is studied, if the therapy is shown to be effective, we then start to see other trials that are designed at the lower age group below that and the larger age group above that. Number one, so that there can be the ability for those individuals on either side of the age range to be part of the clinical trials, but also so that when a company goes forward to seek FDA approval in the US, Health Canada approval in Canada, there's greater clinical experience so that we can potentially get broader approval for the broadest age group possible.
Thank you. And the next question we got was how the truncated dystrophin, I assume from this program, compares to the gene replacement or mini dystrophin from other programs.
The main difference that I can comment upon with the dystrophin that's produced from a gene replacement therapy is that it would be shorter than the dystrophin that's produced from an exon skipping therapy. So the dystrophin produced from a gene replacement therapy is a very, very, very small version of the dystrophin. The one that's produced from exon skipping could be close to the same size, perhaps 2, 3, or more exons shorter than the size of a full-length dystrophin that an individual who does not have DMD would express. So there is potentially quite a significant difference in the size of the dystrophins that could be produced from one class of therapy compared to another. Other questions that are important that are being answered in both clinical trials is how well those partial length dystrophins get to the muscle membranes. And then most importantly for me as a clinician, and I think I speak for many or if not all of my patients and their families, is how does that translate into actual clinical benefit? So once a therapy, regardless of what it is given, it's all fine and dandy for anybody to talk about how it makes the muscle membrane look. That's encouraging. But what's really, really important in the long term is how does this affect the life of that boy or that young man? Does it allow the progression of the disease to slow, significantly compared to what it would be otherwise without them having received that therapy?
Thank you. And I see 2 more questions, and I know we're kind of nearing towards the end, but we got the question that if the question is worded as if out of frame occurs when this was administered to healthy subjects, muscle damage could occur. Did any adverse events occur that would be associated with muscle damage?
I think my interpretation of that is that we're talking about the dosing to the healthy human volunteers.
Yeah, that's my interpretation. And if this person wants to, they typed it in anonymously, but they want to clarify that, please do. But yeah, that's how I took it.
I'm going to answer as to the best of my ability based on my understanding of it. Please give a follow-up question if needed. But the key item that is of concern when healthy volunteers are dosed is, are there any safety issues? So it's normal for any drug trial for healthy human volunteers to be dosed as part of the phase 1 part of that trial. So the first in human dosing, and it's looking for any side effects that may be seen, either anticipated or unanticipated. What I can say from that is there was no reports of there being any significant muscle damage. So there was certainly nothing to suggest that there was any rhabdomyolysis, which is excessive muscle breakdown, significant pain, weakness on the part of any of those individuals. Now, what the study was not aimed to do was extensive biopsies or extensive search to look to see if perhaps a small proportion of those muscle fibers in those healthy human volunteers maybe actually showed some minor dystrophic changes like we would see in an individual who had very, very mild Becker muscular dystrophy. Theoretically, it's possible, but that was not the goal of doing the healthy human volunteer study. It's to look at, are there any safety considerations that need to be looked at? And it was very reassuring in that regard.
Okay, Thank you. And then, Lisa, can I ask one more question?
Sure, go
So
ahead.
up to the last minute here. So another question we received is, what mechanics are used to check that truncated dystrophin is reaching the heart?
The main way that that's being studied as part of this clinical trial and most clinical trials is outcome measures or testing to look at cardiac function. So ECG or electrocardiogram, echocardiogram. That's doing an ultrasound of the heart to monitor the ejection fraction. So that's being done, one, to make sure that there's absolutely no safety issues that arise with regards to the heart, and there's not been any concerns that have been noted on the study so far. But B, over time, to ask the question of does the cardiac function remain stable? Similar to the analogous question in skeletal muscle, does timed functional tests remain stable? The heart tissue obviously is not a tissue that's amenable to biopsy. So nobody's going to go in and take a piece of cardiac muscle to see what the actual dystrophin expression is like and how it might be changing the expression there. And as a result of that, we have to rely on non-invasive measures such as ECG, echocardiogram, in order to be able to monitor in that regard.
Thank you. And that's all I see from my end. And thank you so much for this opportunity. I just want to say, if anyone out there has another question that we didn't get to please email us at community@pepgen.com or clinicaltrials@pepgen.com or Defeat Duchenne, and we can answer more. So I'll let Lisa conclude for today.
Thank you so much. This was extremely informative. As I mentioned at the beginning, we knew that it would be. So I thank you so much for being with us today, and also for sharing with us your time in responding to the questions. I know that that means a lot to our participants. So really appreciate you doing that. And just for those who attended, thank you for coming. Thank you for taking the time to be with us today. Please visit us at defeatduchenne.ca for lots of great information. We have some tremendous emergency care resources. We have a clinical trial finder that's currently going through some updating and some revisions to make it as user-friendly as possible. Follow us on social media. And if you're interested in staying up to date, you can visit our website and sign up for our e-newsletter. And if you're interested in supporting us, please do so. We have our Walk to Defeat Duchenne coming up on Sunday, May 26th. We'd love to see you there. You can find that online as well at defeatduchenne.ca. And just thank you all very much again. Thank you so much, Dr. McMillan, and so much, Elena. We really appreciate the wonderful information you shared and the time that you've spent with us this morning, and I'm sure our attendees do as well.